Hello, my name is Daniela Brits. I work at the Tuberculosis Research Unit at the Swiss Tropical and Public Health Institute in Switzerland. I would like to welcome you to this webinar in which I will go over the main bioinformatics steps associated with short-read sequencing, giving some emphasis on aspects which are important in the analysis of Mycobacterium tuberculosis complex sequences. I start with a workflow, and we usually start with a sample collection at the clinics. And after that sample collection, we typically culture our bacteria and we do a DNA extraction. More and more people are trying to sequence directly from sputum, but still that remains quite challenging. So in most cases, our DNA still comes from a lab culture from a clinical sample. Once we have our culture, we proceed to prepare our genomic library to be sequenced. Here we will be talking only about Illumina sequencing, and if you want to understand how Illumina sequencing works, I encourage you to watch the video which we have posted before this webinar. Once we have our sequencing, we proceed to the bioinformatic analysis. And here I have split this bioinformatic analysis in 4 categories, which is data cleaning of the raw sequences, mapping our sequencing reads to a reference genome, doing our variant call, and then annotating those variants. Basically, these are the 4 main steps which I will be covering in this webinar. Once we have our variants, we proceed to downstream analysis such as drug resistance, molecular epidemiology, or phylogenetic analysis. In reality, these are the analyses that allow us to answer our research questions. They will be covered on day 3 of this workshop by Galo Goich and Christopher Stritt on a few webinars and hands-on tutorials. Let us assume that our sequencing worked well. I will not go into the details of how the Illumina sequencing works again, but again, I encourage you to watch the video previous to this webinar. Sequencing reads are stored in files in a format which we call the FASTQ format. And those files are the output of our sequencing machine. I'm showing here the example of the Illumina HiSeq sequencer. Each of these files can contain millions of reads, and typically, if we have several genomes, we generate gigabytes of data. For that reason, data is typically stored and processed in computer servers. And here I'm just showing in the image of one. So raw reads are stored in FASTQ format. Let's look a little bit in more detail at what this means. Here is depicted how the sequencing information of one read is stored. This information is used by our bioinformatic algorithms in order to process the read in one way or the other. Let's see how this information is used. So to start with, we have the read identifier, which you see here inside this rectangle. The read identifier is composed by an ID and the @ symbol. Following the read identifier, you have a string of characters that can have different meanings and can look a little bit different from the one I'm showing you here, depending on the sequencer used. But for instance, here you can already extract the information that our read has 93 base pairs. On the following line, you have the nucleotide sequence as determined by our sequencer. And in the line below, there is a very important string of characters that allows us to know how confident can we be that each of the 93 nucleotides determined by the sequencer is correct. This is called the quality string and is encoded in ASCII. So a FASTQ file has millions of entries similar to the previous one. And that corresponds to millions of reads that cover our genome. If you would open a FASTQ file in a text editor, it would look like something like the one I'm showing you here. So these are many more reads, not just one. You see that they start with the @ symbol. And if you follow each of these read IDs, you see that the string of characters that follows them is not exactly the same as the one I have shown you before. As I said, that depends on the sequencer. So they are, in the end, slightly different formats of FASTQ files. But all of them contain the read ID, the nucleotide sequence, and the quality string. So next, I want to introduce the concept of pair versus single end sequencing, which is a terminology which you will encounter during this workshop. So one of the preparation steps before we do our sequencing and before we do our genomic libraries is to fragment our DNA into small pieces. So imagine that these gray bars are the DNA fragments which we want to sequence. We can sequence them in two ways. We can do pair-end sequencing, and that means that we will sequence from both sides of the fragment with a forward and with a reverse read. That's pair-end sequencing. Or maybe we choose to sequence just single-end sequencing. That is to say, we only have a forward read. Okay, so there are two types of reads, single-end and pair-end, depending on how you have sequenced your genomic libraries. Usually the pair-end, you can recognize them because they have _1 or an _R1 or _F for forward. So they usually have the same name and _1 and _2. So let's imagine we have sequenced our genome. We have millions of reads, of short reads. They can go from something like 50 base pairs up to 150 base pairs. So what do we do with those? There are two main things we can do. One is de novo assembly, and the other one is resequencing. And I will explain both of them in the next couple of slides. So what is de novo assembly? De novo assembly attempts to build a complete chromosome by assembling these short reads into longer ones, which we call contigs. Those contigs can be in turn assembled into even longer ones, which we call scaffolds. If we manage to assemble different scaffolds together, we can build a complete chromosome. However, this process is a little bit equivalent to making a puzzle of very small pieces. Depending on the complexity of the image of the puzzle, it can be rather challenging to complete the puzzle because the pieces are very small. So in reality, de novo assembly can be more challenging than doing resequencing. And most commonly, we do resequencing. And this is what we will be following in this webinar and then in the following tutorial. So resequencing, I'm sorry. When we do resequencing, we align or we map our reads to a genome of reference. And then we identify what is different with respect to that reference genome. Using this approach, it's usually easier to find the right place for each read. However, you can imagine that if our reference genome does not contain all the genomic regions present in our sequenced genome, those reads will be discarded. They will have no place where they could be aligned to. That is to say, with resequencing, although it's easier, we can only interrogate regions present in a reference genome. So the main steps in a resequencing pipeline are the data cleaning, mapping to a reference genome, variant calling, and variant annotation. Let's start with data cleaning. For doing data cleaning, we usually need to assess the quality of our sequencing. And a very commonly used tool is FastQC, is also the tool you will be using in the hands-on tutorial. So with this tool, we can assess the following: the sequencing quality per base, and per read, the GC content, so the proportion of guanine and cytosine should be similar to the proportion of guanine and cytosine in the genome, which we know in the case of Mycobacterium tuberculosis is rather high, around 65%. So we know if we have strong deviations, there is something wrong. Then we'll also look into duplication of reads. Well, read duplication means that the same DNA fragment has been sequenced more than once, and that can be due to the step of PCR amplification of our genomic fragments. And usually these duplicated reads should be removed or they should be flagged. Or we could have overrepresented sequences, which could come, for instance, from adapters or could come from contaminations. So FastQC allows us to look into all these different aspects. And I will not go in detail each of them. There's quite some tutorials online and also in Galaxy where you can look into how to assess quality of sequencing. I just want to focus a little bit on assessing the quality of our base. And to do that, I need to introduce you to the notion of Phred quality scores. As you remember, when I explained how the FASTQ format is, I told you that each nucleotide has an associated quality value encoded in ASCII characters. And this is what you see here in the upper part of this box. Those characters can be translated into quality scores, which is what you see here. These quality scores are encoded in something which we call the Phred quality. And Phred qualities basically tells us what is the probability that the nucleotide that we have determined with our sequencing is incorrect. For instance, a Phred quality of 10 tells us that 1 in 10 nucleotides called can be incorrect. That is to say, we have a 90% accuracy. And this is rather low, and usually we only take bases or nucleotides that have a 20 or 30 quality score, and the rest is excluded. So the higher the quality score, and this is what you see here on the right, the lower is the probability that we are doing a mistake. How do we actually know what are the quality scores? Again, we can use FastQC. This is an example of an output plot of FastQC. And in the x-axis, you have the position of each read. And in the y-axis, you have the Phred quality, which I explained you the slide before. Plotted are the quality scores of all the reads obtained for a particular genome. And as you can see, the quality decreases towards the end of the reads, which is something which is very common in sequencing experiments. As you can see by the error bars, there are many reads that have very low sequencing qualities. Typically, we get rid of those before we proceed with the rest of the analysis. And that is called trimming. So trimming is a bioinformatic step which we use to get rid of those reads or parts of reads about which we are not really confident about. The tool you will be using in the tutorial is called Trimmomatic. We can also not only get rid of pieces of reads, but also we can exclude complete reads, for instance, if they contain contaminations. That means we have sequenced something else which is not our bacteria of interest, or if the reads are too short, or if they contain too many indeterminations. So all of this can be done with Trimmomatic and Kraken. And again, you will apply these tools in the hands-on tutorial. So once we are happy with the quality of our reads, we are ready to align them to a reference genome. And that is called mapping. So mapping is the process of finding a location for each and every read in the genome with respect to a reference genome, which is what I illustrated here. The tool you will be using for mapping is BWA, which is inside SNP. A very important step in this process is to choose what a reference genome is. Very often, people use as reference genome the lab strain H37RV, which, as you might know, was the first Mycobacterium tuberculosis genome to be sequenced. That has the advantage that this genome is very well characterized. In this tutorial, you will be performing a mapping with respect to the chromosome of H37RV. Oh, I'm sorry. However, we will not be using the nucleotide sequencing of H37RV, but instead, we will be using the sequence of a putative organism ancestral to all MTBC lineages. And I will explain this, which perhaps sounds a little bit esoteric, in the following slides. So this is a phylogenetic tree showing the genetic relationships of the main known lineages of the MTBC. If we go from the tips of the tree, you see on the right, to the internal nodes on the left, we get to a point where all lineages converge to one node, which I indicated here with a star. That node is a putative ancestor of all known MTBC lineages. Putative because we cannot really know how that ancestor looked like. It lived many thousands of years ago. However, we can try to infer the sequence of that genome, and that's something called the ancestral reconstruction. I will try to illustrate what is or how one does the reconstruction of an ancestor sequencing. And I ask you to imagine we will be doing that for the first position of the chromosome of H37RV. Okay, let's take position number 1. And let's imagine that lineage 4 strains, which are here indicated in red, have a G. Lineage 3 strains, which are indicated in purple, have an A. And lineage 2 strains, indicated in blue, also have an A. Lineage 1 strains, indicated in magenta, have also an A. So probably the most likely explanation for this mutation is that the ancestor had an A that changed to a G. If we look into lineage 5 and lineage 6, we see that those lineages have a C for that very first position of the genome. And that is also true for Mycobacterium caneti. Mycobacterium caneti is the closest relative to all extant Mycobacterium tuberculosis samples. So what we would infer is that in reality, in the ancestor of all the lineages, for this very first position of the genome, there was a C that changed to an A in lineages 1, 2, and 3, and that A changed to a G in lineage 4. So imagine we do this for each and every of the 4,400,000 something genomic coordinates of H37RV. In that way, we can reconstruct the ancestor sequence of this chromosome. Why are we doing this? Why using the ancestor sequence? One reason is that we know that all the variants we will encounter in our genomic analysis correspond to a derived state. That is to say, these mutations have occurred after the split of all lineages from a common ancestor. So in these two plots, here on the left, you have the number of SNPs, single nucleotide polymorphisms, that we typically discover when we map strains from different lineages to a reconstructed ancestor. You can see that all different lineages, which are here represented by different colors, have more or less the same number of SNPs. On the right, you can see the number of SNPs we recover from mapping to the H37RV sequence. H37RV belongs to lineage 4, as you might know. As you see, the number of SNPs recovered varies quite a bit from lineage to lineage. This is not so important if one is interested in mutations that have occurred in a recent evolutionary time, like, for instance, the mutations that confer resistance to antibiotics. However, if one is interested in mutational events that happened far back in the past history of the MTBC complex, we should be using the reconstructed ancestor. Now, I told you that the raw reads coming out of the sequencer are stored in FASTA files, and we align those reads to a reference genome. Once we align those reads to a reference genome, that information is stored in files which we call BAM files, because that's called the BAM format. So what is the BAM format? BAM stands for binary alignment map. And that file contains information about, for instance, the position of the read on the reference chromosome, the orientation of the read, whether it's for the reverse, or the quality of the alignment. Here, I'm showing you a graphical representation of a BAM file. And I have opened this BAM file with a genome viewer, so a small program that allows you to visualize read alignments. This one is called IGV. So in the upper part, you can see the reference genome, and you can see the aligned reads to that reference genome. These are these gray bars, and you can see here, for instance, a snip. So this is a base pair, which is different from the reference. So the reference had a C, and our reads had a T, and that occurs at position 761, 155, and actually that's a drug resistance conferring mutation in the RBOBG. Now, how do we know if our mapping, our alignment to the reference genome is good or bad? There are some mapping statistics. So there is something called the mapping quality. And other statistics are, for instance, the depth, the coverage depth. And for instance, how many times a read has mapped exactly just once, or how many times a read has mapped multiple times. Just one word on coverage, because that's something that you will be hearing a lot about. So coverage depth has to do with how many times each base has been sequenced. So how many reads support each base of our genome. To be confident in a base call, each position should be covered many times because, as I said, there is a little error rate introduced by the sequencer. So you have here several examples. Here, let's imagine we have here a variant in each of these red bars. Here we have a coverage depth of 4, a coverage depth of 3, in this case, a coverage depth of 10. So usually we take as a minimum 3 or 4 reads to cover our variant, or otherwise we don't really consider it. But that differs from study to study. So there is no really fixed threshold. So once we have our mapped reads with respect to our reference genome, what we do next is that we try to identify our variants. What is different from the reference genome? This is what I'm trying to illustrate here. The tool you will be using in the hands-on tutorial is snipping. And snipping basically just tries to identify what is different from your reads with respect to the reference. So here I'm showing you a snip, single nucleotide polymorphism, but indeed we can detect other types of variants, for instance, large deletions. So the plot on the left shows you several genomes. Okay, this is what is indicated with these bars. And the different peaks are the reads aligned to our reference genome, which in this case is here in the bottom. You see, so here, this is an indication of the coverage. You see that some genomes have very low coverages for some regions, like these two here, or like these two here, indicating us that these genes, which you see here in blue, which are in our reference genome, are deleted most likely from the genome, from these two genomes which we have sequenced, but not from these ones. You see there is plenty of reads aligning to those two genes. The same rationale applies here. This genomic region is deleted from these two genomes with respect to our reference, but not in these two. We can use the opposite argument to look into duplicated regions, which is to say regions that have a very high coverage, has to say have many reads that align to those regions, can be an indicator of duplications. Duplications are a little bit more difficult to identify than deletions because there are repetitive regions in the genome which are not necessarily duplicated, but would give you the same kind of scenario, is to say a very high coverage. So I said raw sequences are stored in FASTQ files, aligned reads to reference genome are stored in BAM files, and genomic variants are stored in VCF files. So VCF stands for variant calling format. And these files are usually a little bit smaller than BAMs and FASTQs because they do not contain the information for the complete genome. So typically, they only contain the variants. So only those positions for which there is a difference between our genome and the reference. So the way you would read this file is you first have a column which indicates the chromosome. This is not very important in the case of Mycobacterium tuberculosis because we only have one chromosome. This column is followed by a column which indicates the genomic coordinate where the variant sits. So in this case would be the coordinate 3253. Then you have the reference base, which is in this case a cytosine, and then you have the alternative allele, so the mutation, which is in this case T. That's the quality of the sequence of that base. And then you have a string of characters that gives you other types of information like the depth, this is the coverage, which you see here, for instance, here in red, and other types of information, for instance, like the allele frequency. PCF files might differ a little bit depending on the variant color program or algorithm you are using, but they all contain more or less this information. And I want to pick up on the allele frequency information and discuss a little bit with you the concept of fixed versus variable SNPs, which is something which I'm pretty sure you will hear more and more about. So, what are fixed versus variable SNPs? Imagine you have a population of bacteria, drug susceptible, growing inside a patient. This is what you see here on the left. This patient starts treatment, and there is a small population of drug-resistant bacteria that starts to grow. With time, if this patient continues under antibiotic treatment, the antibiotic-resistant bacteria, they will start to take over the entire population. So let us now imagine that we obtain sputum from this patient at different times during his treatment history, and we extract DNA and we sequence. What would we see? So on time 1, we would see no drug resistance conferring mutations. On time 2, some resistance conferring mutations could be appearing together with drug susceptible alleles, and this we could be calling a variable SNP. Sometimes these variable SNPs are also called heterozygote or heterozytes, which is a nomenclature that is inherited from diploid organisms. Formally, that's not completely correct because bacteria are haploids. So that's why we call them variable SNPs. So in reality, this is how our sequencing would look like. At time 1, we would have only the drug susceptible allele, which is fixed. There's 100% G. At time 2, the small population starts to emerge that is drug resistant, and we see the drug resistance conferring allele showing up in our reads. But definitely, this is still a minority of our reads. Once we come later in the treatment and we sequence, we see that this allele starts taking over the population, and in reality, the susceptible allele is now the minority. So, what kind of thresholds do we use to decide whether we call a SNP as fixed or as variable? What I have shown you before was really a simplification. In reality, it's not always very easy to decide when alleles are fixed or not. And the reason for that is that our sequencing methods have some error associated. So some mutations, and in particular, if they appear in very low frequencies, might be due to sequencing mistakes and not due to a true mutation in our bacterial population. So in practice, what we do is that we usually set some arbitrary thresholds, allele frequency thresholds. So mutations that appear at very low frequencies, let's say from 5 to 10%, we ignore because we cannot distinguish them from sequencing mistakes, and we consider the allele in the reference genome. On the other end of the spectrum, for mutations that appear at very high frequencies, let's say over 90, but again, these thresholds depend on the studies. Some people consider it already over 75%. So for those mutations, for which are supported by the majority of our reads, we say we have a fixed variant. And from all variants in between, we usually say there is a variable SNP. This is especially important in the context of drug resistance, although, of course, this can happen with any mutation. However, that can underlie the fact that phenotypic and genotypic DST do not always coincide. So according to the phenotype, the population is susceptible, perhaps because there is only a small proportion of drug-resistant cells. However, when we sequence, we capture that very small proportion in our sequencing reads. Another feature of sequencing analysis of Mycobacterium tuberculosis are the repetitive regions, which are around 10% of the genome. So there are approximately 10% of regions in the genome that are very, very similar to each other. And that means that it's very difficult to find where each of our reads actually exactly maps or aligns. And this is what I'm trying to illustrate here. You have here three different scenarios, the red, green, and blue. In red, you have two genes that have duplicated and are basically 100% similar. In green, you have two genes that have diverged slightly and are only 98% similar. And in blue, you have genes which are 70% similar. Here you have our reads. You can imagine that in the red scenario, it's difficult to find the exact location of each of the reads. It could be gene X1 or it could be gene X2. In the green scenario, it might be slightly easier if there is already some mutations in some of the reads. However, it's still going to be quite difficult to know exactly where this read is aligning to, whether if it's Y1 or Y2. In the blue case, it's much easier to know exactly where is our read aligning. So repetitive regions, they fall into this scenario of red and green genes, which I'm showing you here. And that means that if we sequence those regions, which are equivalent to these ones, with short Illumina reads, sometimes it's impossible to know where is the exact location of each read in the genome. So what we do very often is that we exclude those genomes, which we know, we exclude those genes and positions from the analysis. And these genes are PBB genes, which are a very big gene family, and insertion sequences such as IS6110 or other mobile elements. And again, it's good practice to remove these regions to decrease false positives. You will also be doing that in the tutorial, in the next tutorial. So now let's assume we have our variants. The last step of our pipeline is to annotate those variants. Annotation means we want to classify those variants and the genomic region in which they fall in terms of functionality. One way we annotate is to compare our genomic variants to a file that has described all the genes on our reference genome, and this is what I'm showing you here. I'm showing you part of that file. In each of the lines, you have the attributes of a gene or of an intergenic region. For instance, let's look at the first file. We have a gene that goes from coordinate number 1 to coordinate number 1524. That gene is 1524 base pairs long. It's on the forward strand. It encodes 507 amino acids, and this protein has a certain ID, a pig ID. It's the gene DNA A, and its H37RV equivalent is the RV0001. So you have that for all the 3,989 proteins of the H37RV chromosome. So any variant following, let's say, for example, inside this interval will be annotated as belonging to the DNA A. We can complicate things a little bit and add new annotations to this file. For instance, usually we are interested in knowing whether certain genes are possibly associated with drug resistance. Or I also added here an annotation, which is, are these genes essential for the in vitro growth of H7RV or not? So adding new annotations is very easy. You just need to add them to the file I have previously shown you. So to finish, I just want to give you a general overview of this webinar. So we start with sequencing reads, which in their raw state are stored in FASTQ files. Those sequencing reads are mapped to a reference chromosome. And once they are mapped, that output is stored into BAM files, which contain information about the alignments of our reads with respect to the reference genome. Once we have that, we use bioinformatic algorithms to determine our variance. So what is different from the reference chromosome, which you see here illustrated with a C. And this information is stored in VCF files. So once we have VCF files, that is to say, once we have our genomic variants, we can build alignments and snip lists and start answering our research questions by doing phylogenetics, or by looking into mutations that confer antibiotic resistance, or by doing species determination. So this will be covered in the day 3 of this training. I would like to finish by thanking you for your attention and by thanking my colleagues which have contributed to this talk in one way or the other. Thank you very much.